Hello everybody. Today my topic is on evolutionary neural networks applied to QSR datasets in predicting the IC50 values. So in this work we have applied evolutionary neural networks to find the best solution in terms of fitness function that will select the best architecture of neural networks suited for modeling the biological activity value. So normally designing neural networks poses a problem due to the design of the hidden layers and nodes in each layer. ENNs used to function with the help of operators involved in the genetic algorithms, for example, reproduction, selection, crossover, and mutation. With machine learning being applied to various drug discovery strategies and problems, we believe that the ENNs could be applied in predicting the biological activity values. For example, the inhibitory concentration of QSR datasets. This work suggests that the ENNs are the better predictor of biological activities other than regression and neural networks. So, our main aim of the paper is prediction of the hidden layers of the artificial neural networks to perfect the architecture of the neural networks, to get a perfect architecture. So at first, we need a dataset. So for dataset, we selected TIBO, that is tetrahydroimidazobenzodiazepine. We downloaded the dataset using the Bioloom software from the BioByte database. This dataset consists around 88 compounds, of which 7 were outliers, and 81 compounds are selected to generate a QSR equation. There are 4 indicator variables which have been selected, that is the log P, the beta 18, and the IDMA, IS, sigma. These are the molecular properties which have been selected and been calculated, and the R-square value is obtained around 0.87. The correlation coefficient of 0.932. So this result are been there in the website of the BioByte, and we obtained this. So now our problem is to make the correlation coefficient close to 1 with the R-square value more than 0.94 and 0.95. So to get a proper idea of the biological activity values. So we'll apply the neural nets and probably the evolutionary neural nets to get this value close to 1. So for this we need a test and a train set. Since for neural networks we need this problem to get sorted out with the help of test and train, we divided the 81 molecule set into 3 sets of randomly 34, 35, 44 and 66 molecule set. So for the neural networks, a training set and a test set is required to validate the architecture of the neural network. So I've taken 35 as the train set and 44 and 66 as the test set, again as 44 as training set, 35 and 66 as the test set, and so on we did, and this is the procedure for validating the network of the architecture. So the regressions are performed on the different data sets of 35, 44 and 66. The values are obtained. Actually, regression means about the linear relationship between the actual value and the predicted value. The actual value indicates the experimental value, and the predicted value which have been mathematically modeled with the help of the descriptors value. The correlation coefficient must be sufficiently high if we are to assert that a strong linear relationship between the measured variables. So when MLR is performed in the 3 datasets, the R-square values which are been obtained are given. Is for 35 set it is around 0.86. For 44 dataset it's around 0.72. For 66 it's around 0.81. Now for the 81 dataset molecule, we predicted with the help of neural networks some architectures. Though there are several architectures you are seeing, we have selected only 3 architectures. It was really time consuming while selecting these type of architectures. We have selected more than 30-35 architectures, but among them only best are being represented in this table, and so it was really time consuming for selecting that perfect architecture. So with the help on the next slides we will get to know about the ENNs, how they helped us to find the perfect network architecture of the neural network to get that correlation coefficient close to 1. So the columns or marked in orange indicates the squared correlation coefficient, the correlation coefficient values, and these values indicates that the neural networks perform really better than the normal linear regression equation. These the calculations are being performed on molecular data modeler. So now as we have the training and the test set, we also tested with the train and the network architectures with the train and the test set. So the network architecture is selected is 5591, which indicates that the 5 indicates at first indicates 4 variables with 1 bias term. Next layer the 5 indicates 4 variables with another bias term, and 9 indicates 8 nodes with 1 bias term, and 1 is the output, which is the correlation coefficient. The network architectures are being tested with the 44 and 66 test set, and it has been trained with the 35 training set, 44 training set, and the 66 training set. So various architectures are being tested. Among them, these are the architectures which have been formed are the best architectures with the perfect correlation coefficient. Moving into the next slide, we need to predict the perfect neural network architecture. So what we did the evolutionary neural networks. The steps for the evolutionary neural networks are that initially it was taken 451 architecture, which is the seed of the neural networks. From this 451 architecture, new networks will be generated with a certain fitness value. So at 4, the 4 term indicates the number of variables, 5 is the number of nodes in the hidden layers, and 1 indicates the output. What the evolutionary optimizer does, it selects 100 randomly selected 451 neural network architectures, and it runs for 200 epochs with a mutation rate of 0.05 and crossover rate of 0.20, and with a learning rate of 0.5. The new networks which have been generated were assigned a fitness value, and the networks are run until when it approaches a value of 100. But due to computational power, the networks are run for just 200 generations to get that architecture, because due to time consumption, because it takes a lot of time to compute the network architectures. Then what happens if the optimizer selects the fittest networks from the old parent generation and are mutated with the probability of 0.05 and crossover with the probability of 0.20 to produce new networks. These new networks are again checked with the parent networks if they belong to the fittest generation. So if they belong to the fittest generation, those new networks were passed on to the new generation, and from that a new generation of 100 sets are being collected. This is the form how the our optimizer, that is Pythia, which has been the evolutionary optimizer which has been used in our in this paper. So ENN was applied with the help of the Pythia evolutionary optimizer with the help of another n equals to 81 data set which has been applied, and this network architecture which have been formed, which have been shown in orange columns. So here you find out that the correlation coefficient is almost similar to the neural network architectures within 81 data set. It is almost similar, and the values are almost similar, but the result changes when we test with a different the test and the train sets, and we get very good results than much better than the normal neural networks. So for the again we do the 35, 41, 66 tests have been performed. Among them, the train set we selected the 44 as the test set, and for 35 train set again 66 train set, and the network architecture which have been obtained with the correlation coefficient which have been obtained, and the number of neurons are been given. It's less than 20, but other than that, in the previous neural networks it was much more than 20, and it was the correlation coefficient was very high. So this test have been performed, and for the networks which have been presented, they have been validated with each three randomized molecules, such as molecules of the sets of 35, 44 and 66. That is, a network architecture of 45671 is tested with a 44 set 3 times. This 44 set is been randomized 3 times and been tested with the network architecture of 456, uh, um, and each testing resulted with a correlation coefficient of only 0.93. So this indicates the validity of the network architecture. So at the end, what do we conclude? That the proper networks could be modeled with the evolutionary neural nets, and best network could be predicted along with the hidden layers and the number of neurons in the each layer with ENN. Since here 100 generations are used, the fitness values ranges from 70 to 76, which give good squared regression values if networks are made to evolve around 500 generations or more to get a fitness value of around 100 fitness. So I have said earlier that we have run for only 200 generations due to the lack of computational power. For 500 generations, it would take around 3 to 4 days or maybe more than that to get fitness value. So it is better to develop a neural network in evolutionary way to predict the activity of molecules than normal linear regression methods. So to make the research faster and to get that perfect architecture of the neural network. Other than that, the other fields are other machine learning methods have been applied, and that's the Hopfield networks and the RBF function, which could be implemented with the evolution strategy, which could predict better in different sets of the inhibitor molecules. And this I would end with the next slide, and hope you would like this my paper, and thank you all. See you again.